Investigate Ediacaran community assembly. So Ediacaran organisms occupy one of the most crucial transitions in the history of life on Earth, occurring at the end of the Proterozoic, which is dominated by microbes, and just prior to the Cambrian explosion of modern animals. But it's very difficult to understand much about them because their morphologies were very unique to the Ediacaran. They're not found elsewhere in the fossil record or alive today. And despite the superficial resemblance to plants, the oldest assemblage at Avalon was found in deep water, so they couldn't have been photosynthetic. However, what you do have is thousands and thousands of specimens preserved in situ. And because these organisms didn't move around, their spatial positions encapsulate their life history. So we can look at their spatial distributions to figure out what biological and ecological processes affected them during their life. So to try and demonstrate what I mean, this is a modern deep-sea chemosynthetic community, and we can see there are hundreds and hundreds of little sea anemones around here. And we can mark out where they were. If we remove the photo, we have their spatial distribution, which we can then analyse. So with spatial analysis, we have three basic cases. We have randomly distributed points, aggregated or segregated points. And random points, in this case fossils, occur when there aren't any biological or ecological processes that are impacting the organisms. Aggregated is closer together and segregated is spaced out. And historically, nearest neighbours analyses have been used to determine this. And with nearest neighbours, you plot the distance from each point, the nearest point on a cumulative frequency graph, and this is the red line. The black line is the model, so what you'd expect by random, and the grey is a simulation envelope. If you have your observed line going above the grey area, you have aggregated points, and below is segregated. However, there are a couple of problems with nearest neighbour analysis. The first being that if all fossils are found within 10 centimetres of each other, you have no idea what's going on beyond 10 centimetres, at 15 centimetres or a metre. The second, probably more important point, is you can't differentiate between different models, and sometimes you can't detect them. Thomas cluster models occur with biological or reproductive clustering, and these can be detected using nearest neighbours. However, if you have an environmental signal, such as heterogeneous fossil models, so here we have an island, but within the island the points are randomly distributed, and that would come out as random on a nearest neighbour model. So instead, we're going to use pair correlation functions. The basic idea is quite similar. You have your simulation envelope, your black line is your model, and your red line is your observed data. But here, instead of taking the distance from each point to its nearest neighbour, you're looking at how the density of fossils increases as your radius increases along the x-axis. And this allows us to detect different types of spatial patterns which correspond to different types of underlying ecological and biological models. So, in the case of Thomas clusters, these correspond to neutral models. So this is where the spatial patterns, the ecology, is dominated by biological processes, such as dispersal. And this is in contrast to niche models, which in this context correspond to the impact by the local environment. You also can get more complicated models, such as Thomas clusters on a heterogeneous background, and this is we're going to refer to as a combined model. And also, if you have randomly distributed points, that comes under the niche bracket. And working out whether a community is dominated by the environment or dispersal can be done in this fashion. So going back to our example of the modern deep-sea sea anemones, you can plot your points, you can measure up what their pair correlation function is, and you can see that it's clearly long-random. And if you want to actually test how good the model fit is, you can use a Goodnessoffit test, which can be thought of in a very similar way to an R-squared value. If you have a PD value equal to 0, there's absolutely no description of the model, and if it's equal to 1, it's a perfect fit. So in this case, we've got a very low PD value. Random models doesn't describe it. This is our environmental model, our niche model, which is created using the density of points, and you can see how the environment is represented using this, which you can look at the photo and work it out for a modern community, but for the Ediacaran we don't know what's going on, and so we can reconstruct it in this fashion. And we can see the model describes a bit of the data, but it doesn't describe all of it. We then start thinking about niche models or biological models, And we, again, get a slightly better fit. We're describing more of the spatial structure, but it's only when we combine the environmental and the biological models that we actually start getting a very good fit to our model here. And this kind of pattern is very, very common in modern communities. Most of the patterns are described using a combination of biological and ecological models. So we've been working on the Avalon assemblage, which is the oldest Ediacaran assemblage across UK and in Newfoundland. And we've identified over 40 sites which have over 100 specimens on, which is enough for spatial analysis. Now, taking photographs in order to capture and map out all these surfaces is really quite difficult. It's hard to maintain accuracy, it's hard to capture all the fossils. So what we've been doing is using a laser scanner to do so. And so the laser is mounted, a handheld device, and then we pass the laser over the surface, and this allows us to capture the fossils to about 40 or 50 micron resolution when we're in the field. So here at Mistaken Point, the fossils are captured really well because it's very high relief. It's a little bit harder sometimes to see at first glance when the relief isn't quite so high. Here's another site, a new site, LC13. But we can still see that the fronds made out here. One of the nice things about this particular scan is we noticed when we were looking at it that there's actually a holdfast disc just beside here, which we hadn't originally seen in the field. But we went back out there and had a look, and we saw it. So the scans can help us to actually identify where the fossils are and what you're doing in a way that photographs can't necessarily. So these are some of the sites we've already scanned. We hopefully be finishing off by the end of next field season. And we've got a variety of different structures. You've got the Mistaken Point down here. We've got some of the Charnwood's surfaces in the UK here. But once we've got these scans, the next step is to mark out the spatial patterns. So here in Spaniards Bay. So this is the section of the surface. It's a low-resolution map because you're looking at about 200 gigabytes per surface, if not more. It's not possible to map it all out at the same time, or to resolve it all in the same file. So instead, what we do is we take a high-resolution bit, impose it on the surface, and if we zoom in, you can make out some of the fronds here. So we've just got, this is a couple of centimetres long, and you can see detail at the micron level, third-order branching in some instances. So you can mark out the fossils, and then do that piece by piece for the entire surface, and we end up getting with our spatial distribution maps. And this is the data we have so far. It's a combination of our scan data and some historical data. And so for these surfaces, we apply pair correlation functions, find out what's going on. So for example, Avalon fractures from Spaniards Bay, it's, oh sorry, these have got a bit mixed up. The random model is 0.01, that's incorrect, sorry. And I don't know what's going on there, sorry. We've got our random model, so that's actually a value of, really quite poor. We've also got a niche model which describes it a little bit better, but not much. But we start getting really good model fits when we start looking at the dispersal models only. And what's quite interesting is that the combined model is actually a worse fit than just using the neutral model. I think the actual value for the niche model is 0.33. And we can do this for all the different taxa. So each line corresponds to a different taxa. I've marked out red where a niche model is the best description, and blue where it's a neutral model. And what we find when we combine all these data sets together is that you can see, just looking at this, it's very much dominated by neutral models. You have very few instances where the environment's playing a large role in the life of the species. If we plot up the Goodnessoffit tests for the best neutral model and the best niche model, this becomes even clearer. So each point corresponds to a different taxon, and each different shape corresponds to a different surface. And we can see again that it's very, very, very much neutral dominated with very few niche models. Got a few strange ones like Charnwood's from Mistaken Point U surface up here. But how does this compare to modern communities? So I did some cluster analyses, and we've got the various different surfaces here. And what I looked at is the proportion of niche models to neutral models to combination models. And you can see all the things here, and the different surfaces here. And in Most of these techniques are currently used in plant ecology. So you have amazing data sets for plant ecology, but very, very little data for modern benthic systems. So it's just an example of a modern sessile systems. However, we're starting to analyse deep sea data, and we can see the same patterns. You've got the modern systems are very much dominated by a combination of different factors, and the environmental factors play a huge role in this. And this is in quite dramatic contrast to Mistaken Point and other Ediacaran communities, which are very much neutral dominated. They're dispersal dominated. The environment does not play a particularly good role, strong role in these organisms. So to conclude, we can use portable laser scanners to map large areas, up to 100 metre squares, to a very high accuracy of about 50 microns. We can then use the spatial analysis to investigate what the processes that govern these communities are, whether it's the environment, a niche model, or whether it's dispersal dominated, neutral. And what we found was that Ediacaran community structure is very, very different to modern sessile systems, which are very much dominated by a combination of different factors, whereas Ediacaran systems are just dominated by a dispersal process with very little impact from the environment. Thank you to my colleagues and funding bodies. Thank you.